I'm Dr. Megan Rippy. I'm a postdoctoral researcher at the University of California, Irvine, and what I want to talk about today is urban pesticide variability and its implications for our sustainable water future. I'll try to keep track of the slides, but sometimes I tend to forget, so apologies in advance. So moving on to the second slide, I kind of wanted to start out by talking about the big picture, looking at worldwide pesticide use, because I think understanding that big picture helps us understand sort of the problem relative to urban stormwater. So the first thing you need to know really is that pesticides are a big business. There's a global market. They're a very valuable commodity, and because of this, they are heavily used. Indeed, by 2007, we could say that worldwide application of pesticides for agricultural and urban purposes totaled more than 2 million tons at a cost of over 39 billion US dollars, and that's in 2007 equivalents. So it's a lot of pesticides. And where exactly are they being applied? Well, this map on the bottom of slide 2 from the Food and Agricultural Organization helps us understand that. The colors on this map are average tons of agricultural pesticides per 1,000 hectares of arable land that are applied between 1992 and 2010. So white colors indicate low pesticide application, and dark blue colors indicate very high pesticide application. And I've sort of circled in red the areas that are big pesticide users, obviously the United States, certain areas of South America, Europe, China, and Australia. And moving on to slide 3, we see that there are consequences of this kind of heavy pesticide use. For instance, widespread global contamination of surface waters, groundwaters, sediments, and soils. And if we just take a US-centric perspective for a moment, and we look at some information provided by the USGS, the US Geological Survey, we see that more than 95% of streams and rivers sampled by the USGS between 1992 and 2011 contained pesticides, a huge number. And more than 50% of groundwater wells in agricultural areas were also contaminated. And given these really poor statistics, it's sort of a logical question to ask, who's to blame? So moving on to slide 4, we see that traditionally we've blamed agriculture. And that's what this plot by Stone et al. In 2015 is intended to indicate. On the x-axis, we have percent of streams with 1 or more pesticide compounds that exceed a chronic aquatic life benchmark. So that would mean that they are toxic to algae, vascular plants, invertebrates, or fish. And then on the y-axis, we have 2 different time ranges, top being 1992 to 2001, and then on the bottom, 2002 to 2011. And it's clear that a large percentage of agricultural streams in the United States have pesticides that exceed 1 or more of those aquatic life benchmarks, but that percentage is stable, if not declining. So this piece of information is one of a myriad that has led us to move away from just blaming agriculture and to think about urban pesticide use being a real problem. And that's shown on slide 5, where I've added the urban information below the agricultural information so that we can see that pesticide contamination in urban streams is really increasing, moving from 53% between 1992 to 2001 to 90% between 2002 and 2011. And this means that a larger fraction of US urban streams are now impaired than agricultural streams, and really the majority of US urban streams are impaired. So what are the pesticides and the herbicides that are responsible for this? So who are the culprits? And in the US, it's generally commercial weed killers and insecticides, so a lot of our lawn care products. And there are a wide range of these, but 3 of the sort of big groups would be the phenoxyherbicides, the triazines, and the polyureas. Phenoxyherbicides were developed in the 1940s, some of the early herbicides. They're synthetic plant growth hormones that kill broadleaf weeds through uncontrolled growth. They essentially grow the plant to death. 2 common compounds are MCPA and 2,4-D. You may be familiar with 2,4-D. It was one of the 2 main ingredients in Agent Orange, and now you can also find it sort of on your shelves as Weed Be Gone. Triazines were developed more in 1950s. And they kill broadleaf and grassy weeds by inhibiting photosynthesis. 2 of the common ones that we'll see are simazine and atrazine. And interestingly, atrazine is banned for domestic use in many countries, an exception being the United States. And polyureas, like triazines, were developed in the 1950s, and they also target broadleaf and grassy weeds by inhibiting photosynthesis. And the key player here would be diuron, which is an herbicide, an algicide, and is often used in anti-foulant paint. So it's really common in the environment because it's used for so many applications. So a logical question to ask now is, does this global story reflect the US story that I kind of just told a little bit about? And unfortunately, we have real geographical knowledge gaps in our understanding of urban stormwater pesticides. We know a lot about the US and a lot about Europe, but many of the other sort of key pesticide using areas we know much less about. And so what I want to talk about for sort of the remainder of this talk is a little bit of work that I've been doing with Australian colleagues to help improve this situation, at least for Australia. So now we're going to move on to slide 9. And this is just an outline for sort of the remainder of the talk, which is really going to talk about a case study of urban stormwater pesticides across the continent of Australia. First, I'm going to talk a little bit about the study area and the stormwater sampling methods, then talk a bit about measured stormwater pesticide concentrations, and I'll compare those concentrations to those from North America and Europe, public health standards, and aquatic ecosystem benchmarks. We'll talk a little bit about dominant pesticide patterns in space and time, and even some possible drivers of those patterns, focusing on pesticide chemical properties and the co-occurrence of pesticides in common use groups. So on slide 10, I'm going to talk a little bit about the study area, which is really the continent of Australia. David actually presented some pesticide data from the, pathogen data from the same study. So what happened is that stormwater samples were collected from 10 catchments that were located in 4 Australian states. 3 of these were in Eastern Australia, so Queensland, New South Wales, and Victoria, and the 4th was in far Western Australia. It's actually the state of Western Australia. And it's important to emphasize that all of these catchments were located in urban areas, so sort of 20 to 100% total imperviousness. So that means we're talking about urban stormwater pesticides, not agricultural pesticides. Regarding study design, most of the stormwater samples were collected using automated samplers that were triggered to sample over a storm duration. So that means we have event mean concentration samples. However, stormwater from Western Australia was collected using grab samples due to logistical constraints. And in total, 27 pesticides were monitored using solid phase extraction and liquid chromatography tandem mass spectrometry. And this includes 2 insecticides and 25 herbicides. So moving on to slide 11, I want to talk a little bit about the pesticide concentrations that we observed in urban Australian stormwater. First thing to point out is that only 19 of the 27 stormwater pesticides we measured were detected in urban Australian stormwater, and that included both insecticides and 17 of the herbicides. All of those pesticides are shown along the x-axis of the box and whisker plot on the right, and the y-axis is pesticide concentration in log micrograms per liter. A couple key points about that plot. First, the box widths indicate the number of samples with pesticide detects, so we have more detects for diuron than, say, propoxur. The whiskers indicate the 5th and the 95th percentile bounds of the data, and all the outliers are shown as stars. So the first point I want to use this plot to make is that overall, Australian stormwater pesticide concentrations were low. They're less than 1 microgram per liter, which is that horizontal line going across the plot. But they could be quite variable, in some cases spanning 2 to 3 orders of magnitude. And getting back to this idea that overall pesticide concentrations were low, and sort of putting it in perspective, if we move to slide 12, we see that Australian stormwater pesticide concentrations were generally below those of other developed countries, including the US, Europe, and Canada. That's those blue symbols that I've included on the plot now. They're the global medians with the 95% confidence intervals. And the exceptions to this are circled in red, which are MCPA, triclopyr, dicamba, and desethyllatrazine, where concentrations were more comparable. Urban Australian stormwater pesticide concentrations were also generally below Queensland public health standards for recycled water intended to augment the potable supply. Those are the green symbols on slide 13. And the only exception to this was MCPA, where 4 samples exceeded the public health standard. And finally, urban stormwater pesticide concentrations were also generally below aquatic life benchmarks for fish, invertebrates, plants, and algae set up by the US EPA. That's all of these red symbols now on slide 14. And the exceptions here were just for 2,4-D, where 1 sample exceeded the aquatic life benchmark for plants, and for atrazine, where 1 sample exceeded the aquatic life benchmark for algae. So really the take-home message of all of this, which is shown on slide 15, is that Australian stormwater pesticide concentrations are surprisingly low, which is good news for the environment, as well as for programs intending to capture and use stormwater to augment potable supplies. However, variability is fairly high and could be a problem for pesticides with more stringent human or environmental health restrictions, things like MCPA, so further monitoring is really warranted. So at this point, sort of on slide 16 now, we've shown that pesticides are detectable in stormwater and that their concentrations can vary over orders of magnitude. So what I'd like to talk about now is whether or not there are any dominant patterns in our stormwater pesticide data kind of hiding within that variability, and whether or not those patterns are primarily temporal, varying by season or year, or spatial, varying by catchment or state. So in order to answer these questions, we use two primary methods, which are shown on slide 17. For our first question, what pesticide patterns are dominant, we use principal component analysis to identify dominant pesticide modes or patterns, and pesticide scores, which really tell us about the strength of those modes relative to each sample we collected. And to address our second question, is the primary source of variability driving those patterns temporal or spatial, we turn to resampling-based statistical models. And the way these work is you sort your data by the variability source of interest. So if we were interested in variability by state, we'd sort everything by state. You shuffle all of your samples within that source of variability, so within state, perform principal component analysis to generate a set of sample scores, and then repeat the whole process 10,000 times. The idea being, if you have strong yearly, seasonal, state, or catchment-specific patterns, you're going to recover your original principal component scores every time. But if you don't, each time you shuffle, you're going to get a different set of principal component scores, resulting in a lot of error in your final resampling-based statistical model. And I'll get back to the results of these statistical models in a little bit. I want to start, however, with our principal component analysis and this question of what pesticide patterns are dominant. So our principal component analysis identified two significant principal component modes that explained about 52% of our observed pesticide variance. And those modes are shown on the plot to the left, with mode 1 on the x-axis and mode 2 on the y-axis. And this is slide 18. Many people have probably seen plots like these. They're called a biplot, but just in case, I want to walk through them slowly. The black vectors that you see on these plots are pesticide loading vectors for individual pesticides, and you can think of these loading vectors as kind of defining your principal component modes. So we have a grouping indicated by the dark blue star of loading vectors for mecoprop, dicamba, MCPA, and 2,4-D, and these are our phenoxy and aromatic herbicides. Diuron, our polyurea, is off on its own, marked by the teal star. All of our trizenes grouped together, marked by the red star, and our substituted uracil bromacil is off on its own, marked by the green star. So this suggests that our stormwater pesticide data is sort of well explained in our modes by these four dominant pesticide groupings. So we can think of our mode 1 catchments as being catchments that have high concentrations of phenoxy and aromatic herbicides and some diuron, and relatively low concentrations of trizenes and bromacil, whereas our mode 2 catchments are those that have high concentrations of trizenes and diuron and relatively low concentrations of phenoxy and aromatic herbicides and bromacil. Another key thing that this plot shows us is all these colored symbols, which are sample-specific pesticide scores. And about each of those scores is a bootstrapped 95% confidence interval that tells us the error about the location of that score in principal component space. And the way you interpret these is the further a score is along a given loading vector, the more of those pesticides are in that sample. So if we look at the red circles, those are from catchment Queensland 2, you can see we have high concentrations of phenoxy and aromatic herbicides, but relatively low concentrations of pesticides like bromacil. So at this point, we've identified some dominant patterns in our stormwater pesticide data. What we'd like to talk about now is whether or not spatial or temporal variability underlies those pesticide patterns. And this brings us back to those resampling-based statistical models. We're now on slide 19. And I've shown the results of two of these resampling-based statistical models on the top, the temporal models, with year on the left and season on the right. And they're oriented exactly the same as this data biplot we've just been going through. And you can see by looking at sort of the output of these models that resampling-based models that resolve only year or season-specific variability do not really reproduce our dominant pesticide patterns. There's really large error about our sample scores. It's actually off the chart. It's off the plots entirely. Fortunately, both of our spatial models, our state-specific and our catchment-specific models, better reproduce our observed pesticide variability. Though you can see there's still some large error about the sample scores in the state model. So what this means is that really our catchment-specific model is our best overall model. Indeed, it explained about 91% of our observed pesticide variance in principal component space. And this suggests that our Australian stormwater pesticide concentrations were varying primarily by catchment. And this has important management implications indicated on slide 21, namely that stormwater pesticide contamination is perhaps best managed at the catchment or watershed level rather at sort of broader levels like the state level. So why might urban stormwater pesticide concentrations be catchment specific? We're now on slide 22. Well, one possibility would be that people living in different catchments just use different products. Unfortunately, we have no direct data for this, but one would suppose that if it's true, then our dominant pesticide modes that we identified through our principal component analysis are likely to reflect associations between pesticides that co-occur in common Australian pesticide products. And if we take a look at those associations using a flow diagram on the right, we see that like does appear to be grouping with like, so this may be true. So how do we interpret this flow diagram? Well, the colored circles indicate products that contain only a single pesticide. So for MCPA, we have 173 products that contain only that pesticide. The thickness of the lines connecting two circles indicate the number of products that contain multiple pesticides. So we have 47 products that contain both MCPA and dicamba. And we can see by looking across, that flow diagram that our main pesticide groups are by and large cohesive, but there are a few crossovers, particularly for like triazines and the phenoxy and aromatic herbicides. Importantly, those are agricultural pesticide products only registered for use in pineapple or sugarcane crops. So we're very unlikely to see those in our urban stormwater samples. So for me, the take-home message of all of this is on slide 23, and it's really that preferential application of common pesticide use groups really could explain why certain pesticides tend to co-occur within some of our Australian catchments, but not others. However, there are other possible drivers of pesticide pattern, for instance, chemistry. We can look at slide 24 to see this. We found that pesticides separated into the same four dominant groups when we categorize them relative to their environmental persistence. It's the x-axis of the plot on the right, and we use fuel dissipation half-life as a metric for environmental persistence here, and their mobility as indicated by the log of the organic carbon partition coefficient on the y-axis of that plot. So you can see the same major groups here, and it's less clear why differing chemical characteristics would drive catchment-specific patterns, but one possibility is through climate, because climate can impact both environmental persistence and mobility of pesticides. And given the really broad geographical range of the catchments that we're looking at, it's likely that they had very different climate conditions. So at this point, I'd like to conclude my talk. I'm now on slide 25. I hope I've convinced you that pesticide concentrations in Australian stormwater are surprisingly low, which is really good news for the environment, as well as for programs intending to capture and use stormwater to augment potable supplies. And I want to emphasize here that I'm not suggesting that people drink the stormwater, it's more that they use it for potable substitution purposes. We also found that pesticide patterns tend to be catchment specific, which suggests that stormwater pesticide contamination may best be managed at the catchment level. And finally, we detected several possible drivers of the observed pesticide patterns, including catchment-specific application of common pesticide use groups, as well as pesticide chemistry, particularly related to mobility and environmental persistence, which may be linked to catchment-specific patterns through climate. So that concludes my talk, and I'm happy to take any questions at this point.
I have a question.
Yeah.
Yeah, that was an awesome presentation. And I was wondering, so based on this data set, you can conclude that pesticides in stormwater in Australia are low, but could you find any other published studies anywhere in the literature that contradicted this at all?
That would show low pesticide concentrations in Australian stormwater?
No.
Oh, no, that would find maybe high concentrations.
High concentrations.
Specific studies.
For Australia. There are...
Oh,
just in general. Broadly everywhere, there are certainly studies that show high concentrations in stormwater as well as in roof runoff in some cases where people do aerial spraying of pesticides. So there are certainly instances where stormwater pesticide concentrations are going to be very high, and that sort of came out in the comparison between the Australian stormwater pesticide concentrations and sort of US, Europe, and Canada. That's particularly driven by concentrations in Europe, actually, where pesticides have been used for extremely long time, and they started to ban things when contamination became a problem and other people started to follow suit. In Australia itself, in terms of publications, there aren't a lot that look at stormwater pesticides. There are some unpublished reports out of CAPM and a few studies that are starting to look at pesticide concentrations in wetlands and in agricultural areas. But the urban emphasis isn't as strong yet. But
the fairly rare data set.
Yeah, and the concentrations vary. In agricultural areas, it's obviously much higher. But in the urban areas, they do identify some of the same sort of problematic pesticides that we talked about. But the other issue is sort of what's measured. Atrazine and simazine tend to be commonly detected. The phenoxy and aromatic herbicides people look for less. So that would be sort of the other problem.
Okay, thanks. That was great.
Can I ask a question? I'm interested in have you done any analysis to link the pesticide concentration against the rainfall characteristics?
Rainfall characteristics. That's something that we are working on, but have not done too much yet. I did a preliminary sort of catchment catchment characteristic principal component analysis to try to see if there were land use or rainfall or antecedent dry period kind of characteristics that would separate out the different pesticide groups. And I didn't have a lot of success with that, though there are at least certain land use characteristics that might matter for certain areas. We have one area that's heavily impacted by groundwater, for instance, that has its own signature. But in terms of rainfall, not much has been done at this point with this data, and I did not have a lot of success in terms of breaking everything out by rainfall characteristics.
Okay, thank you.